welcome back. And this is the time for the last lecture of the day. And in fact, the last lecture of the pedagogical lecture series by Stefan Klumpp. So this is his last lecture on dynamics and economy of molecular machines.
Okay, yeah, thank you very much. And thanks to all of you for being back. For still at least pretending that you want to listen to me. Okay, that's, as you just heard, the third lecture in this series. And let me pull up this agenda again. So we have covered all this, and I told you that today I want to look at, again, transcription and translation with a somewhat different perspective, namely with the perspective of economic decisions of a cell. And I also filled in the something else part down here. So what I will talk about is, at the end, is something that's related to this economic question, namely the question how cell growth and gene expression are coupled. That has to do with molecular machines, but the way we describe this theoretically doesn't necessarily show it that closely, that directly. Anyway, you'll see what this is. And I'll start with this first part on the cellular economy of transcription and translation. And to do that, I'll show you this slide again. So you remember we have the genetic information stored in DNA and then read out by these two processes of transcription and translation that are driven by the two molecular machines, RNA polymerases and ribosomes. And the question, or one of the questions that I want to ask today is, are the rates at which protein is produced or messenger RNA is produced, is this dependent on how much machinery is there? So in some sense, it's obviously dependent on that, because if you don't have the machines, then the genes will not be read out. You will not get any protein. But if we don't ask it in such a trivial manner, then the question is, does it matter for the cell? So you have the machines, but does it matter whether you have more of them or less? Also, you could ask, do different genes compete for these machines? Do you have many messenger RNAs and only one of them will get a ribosome to make a protein? And what you will see in the following is that this is closely related to a question that was brought up this morning by Madan Rao. And if you remember, he talked about this question of self-replication. He said a cell begets a cell. In Germany, we call this omnis cellula e cellula, which is Virchow's version of saying this, which means every cell comes from a cell. Okay? And that's important. If you want to make a cell, then you need already a cell. It's the same as with human babies. If you want to have a human baby, you already need the human parents. You can't build them from parts somehow. And at least as far as we know today, we cannot build a cell from parts, but rather we make cells from cells that are already there. And you can drive this a bit further into the microscopic by applying the same idea to this scheme here. Because I told you yesterday there are some loops back because the machines that drive these processes, they are also products of this process. So if you think about it, how do you make a ribosome? Well, to make a ribosome, you need to make proteins, which means you already need to have a ribosome. Okay, so you have loops here. And well, the ribosomal protein loop, that's the simplest one, and that's the one that will show up most in my presentation today. But there are two others. Ribosome also has RNA parts, and to make those, you need an RNA polymerase. And of course, the RNA polymerase in turn is a protein, so to make the RNA polymerase, you need a ribosome. So this idea of building things from parts versus having already something that replicates, that already shows up at the level, at the molecular level, not only at the cellular level. You have already these cycles of self-replication there. And of course, one of these questions that one thinks about once in a while, because they are the big questions here is, how do you get a cycle like this started? You need to get there somewhere, which is this chicken and egg problem. Yeah, you need to start somewhere, and that's what you have here as well. And that in the last years has developed from a somewhat philosophical question into a really practical question with the new field of synthetic cells. Because people really try to build at least parts of these systems that can self-replicate from parts. So to get such a thing started, such a cycle. Anyway, that's not something I will talk about, but it's something to think about once in a while. Okay, let me get back here. So the question is, do these machines affect how much protein we get out, the availability of these machines, and is there competition? And we will look at this with a model system, namely the system of E. coli bacteria growing at different growth rates. This is, of course, in some sense very specific because it's a specific organism and it's a very limited section of what they do in their life, because we only consider exponential growth. But it has a very nice feature that in that case we can, at least in a reasonable approximation, describe the state of the cell by a single number, namely by the growth rate. Okay, that's what you see here. I think this has already been mentioned in the last days. Typical doubling time for an E. coli bacterium is something like 20 minutes, but that's only true if they are in rich growth medium, in very nutrient-rich medium. If give them a poorer medium, this can go up to an hour, to many hours, depending on what is in there. Okay, and if you grow the same bacterium in different growth media, then these bacteria will look very different. That's what you see here. Here you see two types of bacteria, but the two types both are E. coli. Just one type was grown in a rich medium and the other was grown in a poor medium. And that's something that people starting in the late 50s with this group in Copenhagen have studied in a lot of detail how the composition and other parameters of these cells depend on the growth rate. And one thing they found, and this is just an example, this is mass per cell, or think of it as cell size, that can be not exactly, but in a reasonable approximation, described as a function of the growth rate. So growth rate is a good variable in that sense. We can plot things as a function of the growth rate, and growth rate dependencies have been measured for a lot of parameters. So this is from the big handbook on E. coli. There is a chapter that has all these tables, and what you see here is different doubling times, and then you have various parameters. And maybe some of you have already discovered that somewhere one of these parameters is ribosomes. Here, number of ribosomes per cell, and that at this doubling time of 100 minutes is around 7,000. But if we go to a doubling time of 24 minutes, it's over 70,000. So there is an increase by a factor 10 depending on the growth rate. Here's RNA polymerase, and there you also see 1,500 here, 11,000 there. There's also some increase. Okay, so that now gives us a basis to look at these questions. Are the synthesis rates dependent on the machinery and the availability of the machinery? Is there competition? And that's something that we have actually been working on for a long time. And this is the very short answer. Okay, I will not talk that much about RNA polymerase. And it seems that for RNA polymerase, the availability, so the concentration that's really available for a gene to be used, because everything that is transcribing is not available at the moment. So that seems to really depend on the conditions, but the competition seems to be relatively weak. There is definitely competition in stress responses for this sigma factor. That was also mentioned in one of the talks before. I don't remember which one. One of the talks the last day talked about sigma factor. If you don't remember it, ignore it for now. It's not important for this talk. On the other hand, for the ribosome, it seems that the availability does not change that much over the conditions. The number of ribosomes I just showed you changes a lot, but most of these ribosomes are making protein. They are busy. So the ribosomes that are not busy and that could come and start translation of some messenger RNA, this seems to be relatively constant, although there is some discussion. So I had a version of this table without the question mark. I added the question mark because maybe it's not as simple as we thought when I made this table for the first time. Competition, however, seems to be very strong. So if the ribosomes are all busy, or most of them are busy translating, that means there are very few that are free and available to start the translation. That means if you now have several messenger RNAs, then they will compete for these ribosomes. And that's something that will become important later. Okay. That's the second thing I wanted to show you. And this is maybe the most remarkable plot that I have today. So if you don't remember anything from my talk, try to remember this one plot. Okay, what's shown here is on the x-axis the growth rate of the cells. So basically every point here is taken in some growth medium that gives a different growth rate. And then what was measured, and these are several different experiments, what was measured here is the fraction of ribosomal protein to total protein. So the fraction of protein that is ribosomal protein. Why is this remarkable, this plot? Any guess? Yeah, maybe. I mean, what you're saying, that suggests a causation that the cells grow faster because there are more ribosomes, and that I wouldn't be so sure. I mean, it could just be the other way around. The cell grows faster and therefore it makes more ribosomes. I mean, this is a correlation, okay? Let's be careful a bit. This is something you measure, this is something you measure, you plot one against the other. First of all, it's a correlation. And whether there's any causal connection, that's a difficult question. Yeah, let's not get into this. Let's consider this a correlation for now. Someone else did say something which I didn't hear. Someone further in front, no? Okay, that is not remarkable. Well, maybe that is remarkable, but that's actually what it should be. Now, if people do the same experiment, they should get the same result. Okay, there are two things, in my opinion, that are remarkable. One is, look at this point. So up here, 3 doublings per hour, that's a doubling time of 20 minutes. That's about as fast as these bacteria can divide. If you go there, and you take this line, okay, there's no data point, but assume there is another data point up here, then 25% of all proteins are ribosomal protein. That's a lot. Okay, a quarter of the protein that this cell makes is ribosomes. That's something that is really, it's not something I would have guessed. Okay, so fast-growing cells really make a lot of ribosomes. And the other thing that is remarkable, I think, is that this is a straight line. It's extremely simple. You have a linear relation between these quantities. And if you have looked into any biology of that, I mean, ribosome synthesis is controlled in many, many ways. So this is actually something very complex. So you have something very complex that, however, gives a very simple relation. I think that's the other thing that's remarkable. Okay. Yeah, but don't forget this. Really, a large fraction of what ribosomes produce is more ribosomes in a fast-growing cell. Okay, there is a very simple way of interpreting this, and this has to do with the fact that ribosomes make more ribosomes. Okay, and this, so what ribosomes have to do is, well, they make more ribosomal proteins, but they also make other proteins. And we can put this into basically one equation by saying we have exponential growth. If we have exponential growth, then protein, protein synthesis can be described by this equation for exponential growth. P dot is lambda times P, where lambda is the growth rate of the cells. Well, if lambda is the growth rate of the cells, it's also the growth rate of protein. But then we can also say all this protein is made by ribosomes, so P dot should be proportional to ribosomes, and the prefactor is the rate at which ribosomes produce protein. And if I just rearrange this, I can say ribosome to total protein, so R over P total, which is exactly what we have here. This is lambda over gamma, so it should be linear in the growth rate. And the prefactor here, the slope, should be 1 over the speed at which ribosomes make protein. You see that there is an offset here in the plot, which is not in this equation. I can generate an offset here by saying, well, ribosomes are not all active making proteins, so I subtract some inactive ones, and then I get an offset here. There are more complicated models for explaining the offset, but the offset is not so important. What's important is that this tells us, first of all, it should be linear in the growth rate, and the slope should be the speed at which the ribosomes work. Well, the inverse of that speed. And we can read off the slope here, and if we do that, then we get a 1 over gamma that has some strange numbers that we can read off here. And if we convert this, then this gives us a speed of 23 amino acids per second, which is very close to how fast ribosomes work in bacteria. Okay, so this simple one-line calculation here seems to be telling us something that is not entirely wrong. So we have this important loop where ribosomes make more ribosomes. Okay, just to tell you, this is something we did recently. What I've shown you was for E. coli. Recently, we looked at that in Bacillus subtilis. This is the fraction of ribosomal proteins to total proteins. You see that it also goes up to a large fraction here, and then you have this linear relation with the growth rate. And maybe one more thing that is nice to mention. A colleague of mine, Jörg Stühlke in Göttingen, in the Microbiology Institute, he has a project where they try to minimize the genome of Bacillus subtilis. So what they do is, in many steps, they cut out more and more pieces of this genome to make a minimal cell. And at that stage, they had cut out around 40% of the genome. So here you see a map of the genome where all the colored regions are the ones that are cut out. So you have a strongly reduced genome, and what you find there is that with the reduced genome, these cells grow a bit more slowly, but if you look at this ribosomal fraction, it still falls onto this line, which is quite nice. Because, I mean, this cutting out almost half of the genome, that's a strong perturbation. Well, at least you would think that it is. And it is a strong perturbation in many respects, but not for this. Okay. Now let's come back to this line and make a model for this line that is a little bit more involved than, well, not much more involved, but a little bit more involved than this one-line calculation that we had before. So one can develop a systematic theory for this line by taking this idea that ribosomes have to make more ribosomes, but they also have to make other things a bit more serious and really make this more explicit. So basically ribosomes, the idea is ribosomes have to make more ribosomal proteins, they have to make other proteins, and this is another class of other proteins. And if ribosomes want to make more ribosomes, if the fraction is increasing from 10% to 30%, then that has to come at the cost of something else. Okay, so they have to make smaller fraction of something else. So the fraction of what ribosomes, of proteins that the ribosomes produce, you can also think about the fraction of time ribosomes spend on something. Okay, that's like what you do in your day. If you have to spend more time on studying for your exams, then something else, you sleep less or whatever you do. So that's what the ribosomes have to do. If they're busy all the time, and that's important. If the ribosomes were not busy, then that's not a problem. If they have a lot of free time, then they can just allocate the free time. But the important thing is ribosomes are mostly busy translating. So if they have to translate more of something, they have to translate less of something else. Okay, now the idea is that to grow, cells need both ribosomes and they need other things. And other things at this level, at the simplest level, I call the other things just metabolism, everything that produces building blocks. In the end, things that produce amino acids from which their ribosomes can make protein. And we say, well, growth is proportional to both fractions because you need both. That's the idea. Growth rate is proportional to the fraction of ribosomal protein and is proportional to the fraction of metabolic proteins. And there are some experiments which I don't want to explain now that tell us that there is a fraction that is always constant that doesn't. So they're basically things that you cannot take away from, because that has always to be there. Okay, and then we have a constraint that the three fractions have to add up to 1. So in that case, if we say the Q fraction that's fixed, it's always the same, then the P and R fraction, they have to add up to 1 minus the Q fraction. And depending on what the conditions are, say we have a poorer growth medium, the cell then makes fewer ribosomes and more metabolic proteins because the nutrients are poor and they are less easy to process, so it needs more of those. Then ribosomes have to, well, reduce. Or you could say, well, the cell can't grow fast, which means there are fewer proteins per time to be made, so the cell actually does need fewer ribosomes. And it can spend the same investment on processing the nutrients better or faster. On the other hand, one can make the translation slow. So this is our gamma here. This is still the rate at which the ribosomes work. If the translation for some reason is slow, the cell can compensate that by having more ribosomes, so that the ribosomes can keep up with how fast the nutrients, the amino acids, are provided. And that is something that is shown here by this line. So this is the line you have seen before. If one inhibits translation with antibiotics, then one gets lines in that direction. And all of that comes out of this 3-equation model. And because if we just combine these, we can write the growth rate as a function of the parameters. The parameters are this 1 minus q. So phi max is this 1 minus q, and nu and gamma, which are these two coefficients. Nu characterizes the nutrients in the medium, and gamma characterizes the speed of the ribosomes. And then, well, the two cases where we vary one of the two, either the nu or the gamma, that gives us these two lines here. Okay, so this is a very nice, simple, well, it's a theory that is not simple conceptually. It takes some time and one has to be very careful what is really constant and what is varied. It's a little bit like when you do thermodynamics, where one has to be very careful which parameters are varied and which not. But mathematically, this is very simple. It's just a few linear equations. And there are some more versions of this, and we have done some of them where this is extended and a bit more involved. Okay, what I want to, before I do that, I wanted to mention one thing, because I have in this basically just considered this loop here, the ribosome making ribosome making ribosomal protein loop. There are the others. I will not talk about those, but I wanted to mention, this is the same plot a bit more nicely from a comment I wrote some time ago on a paper by a group from Israel, Kostinsky and Rovini, who wrote a very nice theory, made a very nice theory that includes these loops and show that this gives additional constraints similar to the one that I have just explained, similar to this one. And they also show that this, what the theory then tells us is nicely consistent with the experimental data. So that is a recommendation for reading if you want to. Okay, but what I want to tell you today in a bit more detail is the question, how do we actually get this linear relation? So what I've shown you now is there is this linear relation. It's a correlation between growth and ribosome fraction. And we can understand this by saying, well, the cells have some way of adapting how it uses the ribosomes. Ribosomes are used in a way that when nutrient conditions are poor, the ribosomes will make more proteins that process nutrients and fewer ribosomes. Okay, but what we have behind that is some complicated network. You have the whole metabolism that eventually provides the amino acids, and you have the whole control system of protein synthesis, which is also something complex. So how do we get this simple linear relation from all this complex network that's behind it? And to look at that, we made a model that is, again, relatively minimal. So we made basically a minimal metabolism, which you see down here. We say nutrients go in, they become amino acids, and the amino acids are used to make protein. This is a caricature of a metabolism, okay? But what is important, and this we'll see on the next slide, is that there will be some controls here in this pathway. But the important assumption here is that we have one nutrient that is limiting. And we call this amino acid. It could be a single amino acid while the others are plenty, or it could be all amino acids pooled together. That's not really important. What's important is we have one nutrient that will limit growth. Okay. So this is the idea. We have this pool of our limiting nutrient, the amino acids, and we have this metabolic pathway where nutrients from the environment are, by some proteins which are in this P fraction, are imported into the cell and processed, and then this will produce amino acids. And the amino acids are used by ribosomes to make protein. And now we have two ways of controlling these processes here. And one of them is that amino acids will naturally control protein synthesis. So if we have more amino acids, then protein synthesis will be faster, at least before it saturates, because then charging of tRNAs will be faster, and there will be, it's basically several Michaelis-Menten-like reactions. You have charging of tRNAs, you have tRNAs coming to the ribosome, and so on. So the idea is that the amino acid pool will sort of stimulate protein synthesis. Or the other way around, if the amino acid pool is too low, then protein synthesis will slow down. That's the part that's maybe most obvious. Okay, but then there is a second aspect here, and that is that if the cell has plenty of amino acid inside, then it will not bring in more. Because, I mean, if it just accumulates more and more, then at some point it will explode. Okay, so the cell, if you think of this as import of the amino acids, the cell will stop import if the pool of amino acids is too large. That's this other part here. And now in our model that we had before, that means that these parameters gamma and nu, these are now not constants, but they are functions of the amino acid concentration in the cell. Okay, the gamma, the speed of the ribosome, that will increase with amino acid concentration, and the nu, the efficiency of import, or if you think, or the rate of import, that will decrease with amino acid concentration. And what we're doing now is we say, well, if the amino acid pool is given, then gamma and nu are just constants, and then we have the model that we had before. Okay, so if we have these two fixed, then we can calculate the partitioning of our proteome into these sectors, and we can calculate the growth rate. And that's what we do. So the growth rate will be just gamma of A times phi r, or nu of A times phi p, which we can write as this phi max minus phi r. And then, so this basically, you can think of this as also as a balance of the two fluxes. So amino acids coming in and amino acids being used by the ribosomes that has to balance. Okay, and this is one equation for, either phi r or A, so we can just solve this. We can solve it as phi r of A, or we can solve it for A of phi r. And if we do that, so if we take these, basically these functions, plug them into our three equations from before, now they are not that linear anymore, but they can be solved. We just do this numerically. Then we have this phi r of A that looks like this. This is the amino acids, and this is the ribosome fraction. So if we have few amino acids, the ribosome fraction will be high. If we have many amino acids, the ribosome fraction will be low. Or we turn this around and we take A as a function of the ribosome fraction. So if we have a very high ribosome fraction, then the amino acid concentration will be low because the ribosomes just use up all these amino acids and insert them into proteins. If we have few ribosomes, then this will be high because, well, they still import, but the ribosomes cannot use them so quickly. Okay, that's what we have. Now phi r of A, or A of phi r, and if we take A of phi r and insert that back in here, our growth rate was gamma times phi r, and if in the gamma we insert this, then we get the growth rate as a function of phi r. And that's what I show here. So growth rate as a function of amino acid concentration, which is a function of phi r, and phi r directly. This is this curve which has a nice maximum. And that makes a lot of sense, because if we have too few ribosomes, then growth will be slow, because a lot of amino acids are brought in, well, or could be brought in, but ribosomes can't use them because there are too few of them, so the import will be stopped. So we have enzymes for import that are very inefficient because they're inhibited, because there's nothing to do for them. And that means the cell has made a bad decision in making all these enzymes rather than making more ribosomes. And the same over here, if the ribosome fraction is very large, then these ribosomes will use up all of the amino acids and then have nothing to do, while the import of amino acids is very inefficient. So in that case, the cell should have made more enzymes for import or processing of the amino acids. Okay, so there is an optimum somewhere in here, and you can approximate this by just taking the two limiting cases here as straight lines and cross them. How good this approximation is depends a little bit on the parameters that we put into the model. Okay, so all of this we can in principle do for different parameters. Let me go back here. So I have now varied the amino acid concentration or varied the ribosome fraction, but I have two prefactors here. So I still have a prefactor nu0 that characterizes the nutrients and a prefactor gamma0 that characterizes the ribosome speed. So if we take this nu0, this is a parameter that comes from the growth medium. Okay, if this is irrelevant here, if this is 1, then this is basically the rate at which, under good conditions, the nutrients can be used. So nu0 is the parameter of the growth medium, and that we had to give some value in here. So this curve is for one value of this nu0, so for one growth medium. And we can do this now for different growth media, And if we do this for different growth media, then we will get different curves of this type. Okay, so each curve here is a different growth medium. And for each curve, there will be a different optimal ribosome fraction. And if we take the optimal ribosome fraction, then at least with this approximation here, they are very nicely on a line. What we have here is growth rate against ribosome fraction. That's what the remarkable plot in the beginning was, okay? It's just that this was turned around. Let me turn this around. So this is growth rate, this is ribosome fraction. And if I take the maxima or the approximation for this maxima, then they are on a straight line. So what this model tells us is this straight line is basically an expression of the fact that the cell makes this allocation, what is used for the metabolism, what is used for the ribosome in an optimal fashion. If you do it in an optimal fashion, in the sense that the growth rate is maximal, that it can grow as fast as possible under the conditions, then you end up on this straight line here. Okay, so there is one more question now, then that's the question, how does the cell choose this maximum? I mean, obviously, somehow evolution, you could say, well, throughout evolution, the cell has come up with mechanisms to pick this maximum because that was a fitness advantage. But if you want to know what is the mechanism, then we're back to the control of ribosome synthesis, because the cell has to adjust the amount of ribosomes to some value. Okay, and that, I said, is very... The graph is inverted. Yeah, that's... You didn't pay attention. This was the graph. This was the graph. And I inverted the graph to have growth. See, this has growth right here. And I just flipped the axis. Yeah, it looks like a mistake, but it is on purpose. Okay. Maybe I should just have inverted the graphs without I made the labels readable again. Anyway. Okay, so the cell has to pick this maximum in some way. And this is done by the complicated way of how ribosome synthesis is controlled. But one thing that we saw is that actually it may not matter that much for this thing how ribosome synthesis is controlled, namely because if two other parameters, these k gamma and k nu, let me go back and see and show you what these are. So if we take these two dependencies on the amino acid pool, there are two parameters, k gamma and k nu, which you can think of as the thresholds at which either the ribosomes become very slow or the import gets inhibited because the amino acid pool is very high. So we have two amino acid concentrations, a low concentration below which translation will become very slow, and a high concentration above which the import will be stopped. And what we saw when we analyzed this is that if these two are sufficiently separated from each other, so one, the high threshold and the low threshold, if they are not too close to each other, then in between we get pretty much a plateau here in the ribosome fraction. So here the ribosome fraction does not depend very strongly on the amino acid concentration. And that means whatever the control of ribosome synthesis does, it has to basically go through this plateau. And you see if it goes through this plateau for one medium, it will also go through the plateau for the other media in that case. So basically any control function that will go through this plateau will give us at least nearly optimal allocation. So maybe the control of ribosome synthesis, even though it's very complicated, very complex, is not needed for this purpose. So a lot of this complexity is probably needed for other things because, I mean, control of ribosome synthesis does not only adjust the overall amount, but it also makes sure that say, the different components of the ribosomes are present in stoichiometric amounts, that ribosome amount is coordinated with amounts of other things, and so on and so on. Okay, so here we get rather robust control. This doesn't tell us what the mechanism is, but it tells us that it's at least not a problem that is impossible to solve for evolution. So you need to separate these two concentrations, and from the numbers that people have determined, they seem to be reasonably well separated, and then any control that will go through here will be very good. Okay, maybe I'll stop here for a moment and see if there are any questions, because I want to shift gears a little bit for the next part. No questions? Okay, then I'll continue. And well, basically, what I told you so far is that availability of molecular machines, in that case ribosomes, that has an effect on global translation. So overall, how much protein does the cell make? How is the capacity for making protein distributed to making ribosomal proteins and other proteins? It's a very global view on how cells use these machines under different conditions. Fast growth versus slow growth, for example. What I want to do in the following is ask if we have a more sort of local view. We look at an individual gene, how do different growth conditions in which we know that the availability of molecular machines changes, how does that affect the expression of one gene? Okay, and if you normally describe gene expression mathematically, you have equations of this type. Protein concentration has a derivative that is given by synthesis rate minus degradation rate, and growth is typically included as a second contribution to degradation, because cells grow and that will dilute out the concentration. Okay, so effectively you get a second degradation term here, which is the growth rate, and very often this term, the actual degradation, is very small compared to the growth rate, at least in bacteria. So you can just say that P dot is alpha minus lambda P. So the steady state should just be P equals alpha over lambda. Or if you multiply this over, you can say protein concentration times lambda should be constant. And that's something that one can look at experimentally, because you just have to measure the concentration of a protein at different growth rates. And this is what you see here. So these are not our data. This is a collection of data from, well, some of my collaborators and some from the literature. And well, what you see here is it's not really constant. Okay, it's reasonably, given the scatter of the data here, it's reasonably constant at fast growth, but at least if you go down to slower growth, then it's not, there's a clear deviation from constant. You could say, well, this is because of the beta zero, so we can put the beta zero in, but that doesn't work quantitatively, because at least with the LAC-Z protein, we know what the degradation rate is, and if we fit this, then we get a degradation rate that is way too high. So this deviation from constant is not explained by the beta zero. So what that means is, well, if it's not that, there are not many options in the equation. That means that the alpha, the other parameter that we have there, also needs to have a dependence on lambda. So alpha is an alpha of lambda, and we have interpreted this as availability of RNA polymerase. That availability of RNA polymerase changes as a function of growth rate. So if instead of looking at this strange combination, lambda times P, we plot the protein concentration directly, then protein concentration, this looks interesting. Let's hope this is the only thing that looks interesting. Protein concentration as a function of growth rate gives this nice decreasing function here. And if we want to include this in sort of network type models of gene expression, then we can basically just say, well, growth is something that reduces gene expression, so we treat it like a pseudo-regulator, something that represses gene expression. And this, I'll show you two examples where we have looked at this. Growth, if we have now a gene that is repressed by a negative regulator, then growth will have a negative effect on the gene directly, but it will also have a negative effect on the repressor. So we have a negative effect and a double negative, so a positive effect. So in that case, a growth dependence should be weaker than if we didn't have the control. And if we have a positive regulator, something that activates this gene, then we have a direct negative effect and we have an indirect negative effect. So here we should get a stronger growth dependence. And that's exactly what is seen. So these are experiments from Zhang, one of my collaborators when I was a postdoc. So this is already quite old. This is the black curve is the unregulated case. With the negative regulation, we get a weaker dependence, and with the positive regulation, we get a stronger dependence here. And this game you can play with all kinds of regulatory systems. And people have done this in quite a few systems. And there is one case that I want to point out because it's interesting, and that is when we not only have an effect of growth on a gene, but we also have an effect of the gene on growth. For example, if the product of a gene is toxic. If a cell poisons itself, then the gene slows down growth, but growth will also reduce the expression of the gene. And you have additional things of this type in antibiotic resistance, for example. And then what you get in all these examples is that growth rate can become bistable. And you have a population in which some cells grow fast and other cells grow slowly. Yeah, here, the cells that don't make the toxin or make very little of the toxin, they will grow faster, but growing faster will also make them make even less of the toxin. So you have fast-growing cells with small toxin concentration and slow-growing cells with higher toxin concentration. Here you see one example where this has been demonstrated with fluorescence. So you have lots of these dark cells, and in that case, the dark cells are the fast-growing ones, and the bright cells are the slow-growing cells, which you can see because there are many more of the dark cells. That's because these have been growing a lot. Okay, I'll come back to this example with the antibiotic resistance.
The process is concentration dependent in this case? The process which you see in the...
Which process?
In B, figure B.
In this one?
Yeah, slowly growing one.
So this is this example where you have basically expression of a T7 polymerase that slows down growth, but so the T7 polymerase drives this fluorescence, but it also slows down growth, and the growth has this negative effect on the expression. So basically, if a lot of this T7 polymerase and the fluorescent protein is made, then the cells will slow down growth, but slowing down growth makes them make even more of these proteins, and then growth slows down even further. So there's this positive feedback between protein synthesis, between synthesis of that specific protein and growth. Yeah. Okay, and one thing, since at this meeting we have talked a lot about stochastic dynamics, let me show you one thing that we did related to this bistability. And this was work by Anjan Roy when he was a postdoc in my group. He's now at IIT-Bombay, I think. No, Delhi. IIT Delhi, sorry. I should know this, yeah. I think he moved there this year, at least not long ago. Okay, so what we try to do there is, there we thought about if we have these populations in which some cells grow slowly and some cells grow fast because of these interplay of the proteins they make and the growth, then actually how we typically simulate the dynamics of gene expression and how the experiments are, and these are two very different things. So what we typically do if we run a simulation, we look at one cell, we look at the dynamics of what happens inside that cell, for example, with the Gillespie algorithm that was mentioned yesterday, I think. Anyway, so you have numbers of molecules of some proteins, and you watch them over time. At some point here, the green and the blue, they switch. Green goes down, blue goes up, and then the cell changes its overall state. But what you really look at is one cell over a very long time. For some time, this is a slow-growing cell, and then it becomes a fast-growing cell, and then it goes back to slow-growing. So you watch one individual cell for a very long time, and you see both behaviors, fast-growing and slow-growing, in this one lineage of cells. That's not what you do in an experiment. In an experiment, there are experiments where you do exactly this, but these are not the simplest experiments. This is something where you have to take specific devices to do that. If you just grow cells in a dish or on a cover slide, then what you do is you take snapshots. You take a snapshot of a whole population, and there will be some slow-growing cells and there will be some fast-growing cells. And typically there will be many more fast-growing cells than slow-growing cells because, they have been growing fast and they have been multiplying. Okay, so, and these two things, usually, this doesn't matter, this difference. It doesn't matter if all the cells grow with the same growth rate. But if the cells grow with different growth rates, and in particular if there's really two very different growth rates, we have this bistable situation, then these two ensembles of how you can look at the cells and the gene dynamics, these are not equivalent. Okay, so what we thought is, how can we simulate this case rather than this case? And what we have to do, first of all, we cannot simulate just one cell for a long time. We really have to simulate many cells in parallel, so that at least we get a picture like this. But then we have to implement somehow this, this growth. And what we do is, we have several cells in parallel, but then we also have in addition a cell division. So cells from time to time will divide into two cells. And when they do that, we could of course simulate all cells, then this will grow exponentially and we would run out of computer time very quickly. So there is a way to avoid this, and the way is to throw out some cells. And if you have this single cell that you simulate for a long time, you could imagine that that's exactly what you're doing. You simulate one cell, this cell will divide from time to time, but you only keep one of them. Okay, implicitly that's what you're doing all the time. You ignore the cell division because you throw out the second cell every time the cell divides. But and this we can do, of course, also in parallel with many cells. Then we have something that is, if you have heard of this device, the mother machine, where you look at individual cells dividing in parallel, that would be what we simulate if we have this with many cells in parallel. If you haven't heard of this, then ignore it. But now we want to simulate this case where the cells don't, where you don't have one cell. And what we do in this case is we still have to throw out some cells, but after every division, we throw out not one of the daughter cells, but we throw out a random cell from the population. That is exactly the same thing that's done in evolution in the Moran process, if you remember that from 2-3 days ago. Do you remember that? One of the speakers talked about this. I don't remember who it was. Yes. Okay. So that's what we do here. So every time one of these cells divides, so cell division is just one of the stochastic reactions that happens in these cells. And every time one cell divides, we throw out a random cell. Then we get a population that looks like a growing population that you dilute from time to time. Well, that's what people do in the experiment when they want to keep exponential growth for a long time. From time to time, they dilute their population and let it continue grow. So here we do effectively the same thing, we don't dilute from time to time, but rather we have sort of a micro dilution every time a cell divides. Okay, so that's one thing we need here. And then if we want to get back to this, if we want to do this for a reasonably large population, we need an efficient method to do this simulation. And we use something that we call the next family method. And maybe I will not go into this, but the idea is there are different versions of this Gillespie algorithm for simulating stochastic systems. And the trick there is how you count. Basically, you have to always decide what is the next reaction that happens. You can either do this by deciding in every step anew, or you can make a list in which you sort and what is happening after which, what is happening in which order. And sorting can be done by different algorithms that have different efficiencies. And the trick here is a combination of a sort of efficient sorting algorithm plus a grouping of reactions that don't interfere with each other. And since we have different cells where every reaction happens in one cell and not in the others anyway, except for division, so we have sort of a natural sorting that some reactions happen here and don't affect what's there, and some reactions happen over there and don't affect what's here. Okay, so one can come up with a very fast algorithm to do this, so we can simulate large numbers of cells without problems. Okay, let me show you one application for this. This is a system, it's basically a minimal system where we get bistability with different growth rates. So we have two genes repressing each other. So one gene represses this one, this gene represses the first one. That generates bistability. We have either gene 1 turned on and 2 off, or we have 2 turned on and 1 off. And then we just say, well, whenever this gene is on, growth is reduced. This is a bit artificial, but it generates what we want to have, namely a population that has two different states, one which grows fast and one which grows slowly. And then we can simulate either this case where we have a population, or this case where we have a single cell growing over a long time. And we can do the statistics over the concentration of this protein, which I call the toxic repressor because it represses this gene and it also reduces growth. And if the repressor is not toxic, that's the green curve, then whether we do this simulation or this doesn't make a difference. The dotted green line and the solid green line, they are exactly the same. But if we have this growth difference between the two states, then you see what I described before. In the case of a single cell watched over a long time, we get a small peak here for the fast-growing cells, so those with a small concentration of the toxic repressor, and a larger peak here with a high concentration of the toxic repressor. These are the slow-growing cells. So basically, if we look at this for a long time, there will be many of the brown ones and relatively few of the blue ones. While here it's exactly the other way around. When we look at the population, we have a strong peak here for the fast-growing cells, and we have a much smaller peak. Where is it? It's not even visible, it's so small. Okay, there is a small peak here which you can't really see for the slow-growing cells. So here the fast-growing cells dominate because, they have multiplied. They divide more often, and every time they displace one of the slow-growing cells. Well, not every time, but they do displace many of the slow-growing cells. They have more chances of displacing than the other way around. Okay, Here's a more interesting example, and that has to do with antibiotic resistance. This is a system that was studied by Terry War's group some years ago, and what they have shown there is that a gene for antibiotic resistance, this has a positive effect of growth via this, or a positive feedback on it via growth through this loop here, because the antibiotic resistance gene reduces the antibiotic concentration in the cell. The antibiotic concentration has a negative effect on growth. But in that case, because of the specific system, growth had a positive effect on that resistance gene. Okay, so we have a positive feedback here. And what they've shown is that at intermediate antibiotic concentration, there is indeed bistability. So you have some cells like this one that doesn't divide and grow, and you have other cells like this one here that grows and divides very frequently. So you have a mixture of fast-growing and slow-growing cells. Okay, and then we have implemented this in our simulation. And what you see here is, okay, this is a somewhat busy plot. If we have the low antibiotic concentration, then, I mean, the dotted curve and the solid curve, they are almost the same. And there's clearly a strong dominance of these cells that have a high concentration of the resistance gene, so cells that can grow despite the presence of the antibiotic. If we have a very high concentration of the antibiotic, this is this one, the 1000 case, then also the two curves, dotted and solid, they are the same, the red curve here. And this is dominated by cells that don't have the resistance gene, because in that case, the antibiotic simply overwhelms the cell stops making the resistance gene and stops growing. But in the green curves, you see that, I mean, already very clearly here, the green curves are different. That's the intermediate case where we have bistability. Some cells grow fast because they make the resistance gene. Some cells grow slowly because they do not make, or only make small amounts of the resistance gene. And in that case, we have exactly what we had in the slide before. In our single cell experiment or simulation, we would mostly see this peak here. There's a green dotted peak with low concentration. So essentially, we would just see cells that don't grow. While in the population, one would see mostly cells that do grow and a small fraction of cells that don't grow. Okay, so exactly as we expected, at the intermediate concentration where the system is heterogeneous or bistable in growth, we have fast and slow growing. It does matter how we do the simulation, and it does matter how we do the experiment. Although in the experiment, I mean, since this is simpler, typically that's what you would do. But what it also means is that when you use an experiment to measure how these cells react to the antibiotic, depending on which type of setup you have, whether you measure effectively single cells over a long time or whether you measure this population average, you will get different curves of the dependence, how the growth rate depends on the antibiotic concentration. Okay, here you see the dotted line. This is our single cell type experiment. Already at a relatively small antibiotic concentration, these cells will effectively stop growing. While in this population, you have this much more gradual decrease, and only at a much higher concentration, they really stop growing. So if you have some quantity like a minimal inhibitory concentration, which is where they stop growing, or some IC50, the concentration where the growth rate goes down to half the initial growth rate, these quantities would both depend on the type of experiment that you use. So it depends on the setup of your experiment what these results will be. It's something to keep in mind, because, I mean, if you do these fancy experiments, maybe you get something completely different than what you do with a simple population growth on a cover slide. Okay, I think I was fast today. I guess you don't mind. Anyway, so that was kind of a long journey away from the transcription and translation. We started with the transcription and translation. We started with the fact that machinery for expressing genes is, the availability of the machinery is dependent on the conditions. There are some conditions where the cell has more ribosomes or more RNA polymerases, and others where there are less. And that will have an effect on the expression of genes. And what I told you in the beginning is that efficient use, in particular of ribosomes, because ribosomes you can think of as very expensive machines for the cell. And if a cell makes a very expensive machine, then this machine should be used basically all the time. Yeah, this is, sometimes I didn't have it in this slide, sometimes I have a plot, a picture of an airplane in these presentations. And if you think about an airplane, this is an expensive machine. So this should be used efficiently. If you're the airline, then, I mean, that's an expensive investment, and so you have to use it efficiently. And then one thing, for example, is, one question you could ask is, why should the airplane get, say, from Frankfurt to Bangalore very fast? Well, you could say, well, the airline is interested that I get here very quickly. But that's not what the airline is interested in. I think the airline doesn't care whether I get to Bangalore a few hours earlier or later. What the airline cares about is how fast the people can be unloaded and then reloaded, and then you can transport more people that, again, pay tickets. Okay, and that's why the airplane from Frankfurt comes here, arrives in the middle of the night, and then basically immediately flies back to Frankfurt, and then back again every day, and so on. So you have an expensive machine, you have to use it very efficiently. And the same thing is true for the ribosomes. The cell does not care whether this protein is made in half a minute or in 25 seconds. But if the protein is made in 25 seconds, then you can make another protein with the same machine. So you will make more proteins in the same time with the same number of ribosomes. Yeah, so this idea that the ribosome is an expensive machine and that it has to be used very efficiently. You don't want it to sit around and do nothing, and you don't want it to move very slowly, because that's the same as going fast and then sitting around. That is a very useful way of thinking about it, and that can explain a lot of the constraints on this system. And one of the features that I have explained today is this linear relation between ribosome fraction and growth rate. And the essential assumption that goes in there is that the ribosomes are busy. They either make ribosomal proteins or they make other proteins. And this capacity of making something, this has to be used in an efficient way. And I have tried to show you with this model of our minimal metabolism and some control of ribosome synthesis, how this can lead to this optimization that ribosome is really used in the most efficient way so the cells can grow fast. Okay, and then I have somewhat diverted a little bit and shown you this, this view from the individual gene or from the small circuit of genes, where these effects, these global effects of growth can be included as a pseudo-regulator that affects the individual gene. And shown some examples, and at the end I have shown you how we can include these things in stochastic simulations, which is particularly important if populations are heterogeneous. And I mean, typically populations are heterogeneous. The question is whether the heterogeneity is just some, you have a peak with some width, and you say, well, okay, let's ignore the width and let's just say the cells are all the same and they're somewhere in the middle of the peak. But, well, either the peak is very broad, then you might not be comfortable ignoring this, or if you have a peak that's not one peak but actually two peaks because the system is bistable, then you really cannot ignore this anymore and you have to take it into account. Okay, that is everything that I wanted to tell you today. So maybe you take some of these things with you and remember them. The one thing I still wanted to do at the end, I wanted to show you again that behind all of this, at least behind the parts that are sort of research and not just things that you could also find in a textbook if you look for them, behind all of that is of course a group of people, and I wanted to point out a few that were involved in the things that I've told you in the context of accuracy. This was mostly Mamta Sahu. In the context of coarse graining, there was work together with a student, David Seifert, and my colleague, Peter Solich. And in the things today, that started when I was a postdoc in Terry Wa's group at UCSD, and it was in large parts a collaboration with Matt Scott, who is at the University of Waterloo in Canada. And then with the bacillus work, I worked together with Jörg Stühlke in Göttingen and his student Binyao Zhu. And the part at the end was together with Anjan Roy, who, as I mentioned, is now in Delhi. Okay, this is all I wanted to tell you. So thank you very much for listening. And if you have questions, I think we still have lots of time for that. Thank you.
Questions?
Working for these two functions, or two genes which are like inversely correlated to each other?
I'm not sure what you mean with the two genes, but do you mean what generates the toxin and what reduces growth? I mean, it could be the same or it could be different. It doesn't really matter as long as they are produced together. But so the simplest case you can imagine is that there is one gene and the product of that gene either has the function to suppress some important process in the cell, or it has a side effect that it has some negative effect on the cell. Let's say you have an inhibitor of some important metabolic enzyme. So if this enzyme will then, when the protein is expressed, work at reduced capacity, then the growth will be reduced. But yeah.
Is this the only way that growth can affect expression, or can it affect this gene expression in some other way as well?
No, there are many ways. I mean, it could, basically you can affect growth. Well, okay, let's go both directions. So growth affecting gene expression, I think this mostly works via availability of machinery. But you could do it by other ways. I mean, but the typical case would be availability of ribosomes and of RNA polymerases. If you reduce the concentration of ribosomes strongly, then the cell will have a problem making proteins. But in principle, you could also do it via the metabolism. The other way around, it's the same thing. So if you have a gene that is expressed and has a negative effect on growth, it could simply inhibit the ribosomes. That would be one option, but it could also inhibit something in the metabolism as long as this process is important. So if you reduce the import of some nutrient, that would be a way how growth would be reduced. If you slow down DNA replication, that could be a way of reducing growth. So there are many ways how this could be done. It could also be, I mean, it could also be that some protein binds to DNA in random places, and just since once in a while it binds, if you have a lot of it, it will once in a while bind in a place where it shouldn't. That would also have a negative effect on growth. So there are many ways how you could have this. And in the end, I mean, at that level of description, it doesn't matter. If you look more closely, then it might matter, and there might be subtleties that I haven't talked about.
Can you use mic?
You can use two microphones.
I just have a question about this Moran-like process. You showed that this cells you threw out means every generation. So is it that you kept the sample size constant, like 100 cells?
Yes, yes, not 100, but more. I mean, maybe 10,000 or some
10,000.
large number, yes.
And also you showed some plot where there was a Miller unit, so in y-axis. So what is that Miller unit? So I don't know.
Oh, this is a way how, I mean, this has to do with how the experiments are done. So this in that case, the gene expression was measured by the activity of an enzyme, which is this like Z enzyme, beta-galactosidase. And the Miller unit is a way of how you measure the activity of that enzyme. But you can think of it as proportional to the amount of enzyme that you have. So you could also measure the protein concentration. Then you should get the same plot. Or you could, instead of having activity of an enzyme to measure the concentration, you could have a fluorescence reporter that should also give the same thing. So this is a way, I mean, that's something I didn't go into. It's a way how you measure how much protein is made. And this is a traditional molecular biology way of measuring this. You measure how much protein is made by how active a chemical reaction is that is driven by an enzyme that you use to measure how much of that enzyme you have. Okay, so you use the enzyme, put the gene for that enzyme at the point where you want to measure how much of the gene is expressed, and then you look at the activity of the enzyme.
Yeah, in your stochastic simulations of gene expression, you had one system where you had linear progression of cells, and the other where have you population density. So, what is the event you are choosing? I did not understand that. Like, what is the event of that Gillespie algorithm that you choose, for which you had to choose the time?
Yeah, I didn't explain that really. But so what we have is inside each cell that we simulate, we have some genes and we have some dynamics. So mostly we make new proteins and we might degrade proteins. Or we could have something that's more complicated where we describe binding of proteins and proteins activating other proteins and so on. So we could have any dynamics. In these cases, it was really just synthesis and degradation of proteins. That were the processes. Then we add cell division as an additional process. Okay, that's typically slower because it only happens every half hour, while making proteins happens a few times per minute. But so this is added as an additional process, and for that we just run a Gillespie type simulation. But every time a cell divides, we have this additional thing that now we generate a new cell, and we throw out one cell.
Okay.
I can hear you. I'm not sure if this mic is for the recording or for me.
I am telling that the ribosomes which were there, they goes and binds to the mRNA only by simply diffusion, or there are other kinds of process. I mean, any proteins goes and take it to the perfect mRNA. I mean, then the rate will substantially be increased or decreased accordingly.
I didn't understand the last part.
I mean, if you are we are having a pools of mRNA in the cell content. Okay, so there when we are presenting a ribosome to it, so there it is only by simple diffusion, or there are any other mechanisms underneath it.
Well, I think of it as diffusion, but maybe not simple diffusion, because it's diffusion in a complex environment. You have this densely packed cell. The ribosomes also often bind to messenger RNA while the messenger RNA is still being transcribed, so it's still connected to the RNA polymerase, so it's not necessarily a freely diffusing messenger RNA. But yes, I would think of it as diffusion. So there is, as far as I know, nothing that brings the ribosome actively onto the messenger RNA. But then the binding, that's the thing that Debashish Chowdhury mentioned yesterday or the day before, it's not really simple binding of the ribosome, but rather there are the two subunits of the ribosome where one of them binds first and then the other comes and joins it, and then the whole thing assembles to start translating. And also you need additional proteins that help the ribosome binding and starting. So it's a bit more complicated, but the simplest picture, I think, is really you have the two things diffusing, they have to meet, and then the process can start.
Thanks.
He's not looking at me. Yeah. See, what you said, if I understood correctly, the ribosome have to be utilized as an overall. But then cell also has to decide, how it is going to distribute them. That means whether it will fly to Bangalore or go to New York and so on, which has the higher priority, right? So how does a cell, have some kind of a decision-making process here?
Yes, I think that's what you need gene regulation for. Basically, the cell, so in some sense, the cell does not tell the ribosome to which messenger RNA it should go, but rather the cell makes those messenger RNAs to which the ribosomes should go. So if a cell should make more ribosomal protein, for example, then the cell has to make more messenger RNA coding for these ribosomal proteins.
So then the question is pushed back to transcription rather than to translation. Is that a correct statement?
Yes, in principle, yes. I mean, you could also do it at the translation level by regulating the translation initiation, but I think most of it is really pushed back to transcription. So the messenger RNAs compete for the ribosomes. If you think of it that way, the ribosomes is a scarce resource. You make messenger RNAs, they compete, and you bias the competition in a way of making more messenger RNAs of one type than of the other.
But that is also done by some other proteins. Is that right?
Yes, for that you need additional proteins, but not many. I mean, regulators typically have small concentrations. Okay. But yes, in principle, yes, there are additional things there.
So in order that some protein gets priority, some other proteins will also have to get priority so that they can either suppress or increase the expression of the protein.
Yes.
Okay.
Any other question?
Yeah, I mean, if you go to that level, then the whole complexity that we have ignored will come back eventually. I mean, this whole complexity is there for some reason. So once you start thinking about all these redistribution decisions in detail, then all the regulation that is behind it will come in.
Any other question? Okay, if not, then let me again thank Stefan on behalf of all the participants as well as